London, and from
Berlin.
And from 2007 he is in the PTB, Physikalisch-Technische Bundesanstalt, also in Berlin, and he recently was provisionally awarded Ramon Michello Fellowship. Sergio started his research working on stochastic phenomena, and that has been that he has kept in his within his career. He has done a number of different works on nonlinear phenomena, pattern formation, and more recently he has used his knowledge to address a number of problems in biophysics. And I think that's the subject of his talk today, on mechanism of pattern formation in models of the interior of living cells. Sergio.
Thank you. Thank you very much for the introduction. It's a pleasure to come here to Mallorca, because after 8 years in Germany, you are not a real German if you have not been in Mallorca, at least for a weekend. So I'm going back to Germany and feeling much more German now. And anyway, it's also a pleasure to come to this institute, because also during a lot of years I have visited conferences on physics, for instance, and there was always a amount of people from at that time in IDEA, now ePhysics, and it was interesting to come here. And I'm doing this. And here is the, yes, I'm working in Physikalisch-Technische Bundesanstalt. It's a institute about standards and calibration and is in collaboration with the industry, always working sometimes from the industry. I'm a little bit a part of them because I'm getting the fellowship from the German Science Foundation, and with some projects, and then I get my position from there. So they're in PTB, they are working in some nonlinear phenomena which are not really related with calibration of things. So, but it's a nice place also. I recommend you to visit, and they are doing also very interesting things. I'm working in the group of MARCKS there, and you might know also. And that's the title of the talk. I will speak about pattern formation, so it fits very nice here, and in particularly in the models of the interior of living cells. I have some collaboration with experimentalists, also, which I will present in several slides, but the main subject will be more theoretic and numeric. Also because I didn't know that Maxim was going to introduce me, I will present also the kind of research line that have been done. This is more related with my beginning in the University of Barcelona, with chemical systems and stochastic effects in pattern formation. Then part of this reaction diffusion process, I applied to scroll waves, which are three-dimensional structures, so I went to three-dimensional systems where we have a lot of relation with cardiac dynamics. So you can take it more in a theoretical point of view in excitable media or generic excitable media, or go to wave propagation in cardiac tissue, where I have done also, and then you have to look for more complex models of wave propagation with more equations, but the dynamics are sometimes very similar. And there are now also, we are also working in some multi-scale problems of how can instabilities of two different wavelengths, how they can, or characteristic size, how they interact, and what is the importance of this, and how can we control it, also beginning to apply the delay feedback. And also pattern formation in heterogeneous reaction diffusion systems. So the chemical system you consider a continuous systems where the chemical species have diffusion, have reaction, but you can consider also what is happening when there is more heterogeneities inside of the system, or going to discrete reaction diffusion systems, like for instance the cardiac tissue. The cardiac tissue is actually is for mass cells. I mean, typically you go from cell, you homogenize, and then you go to the continuous. But if you stop before going to the continuous, or once you are in the continuous, consider again the discretization, several effects appears, and so I'm working on these kind of things. Today I will focus on what I'm doing more recently during the last 2-3 years, applying nonlinear dynamics to processes inside of the cells. And some applications to active interfaces in the sense that the membranes of the cells are actually an interface between the interior and the exterior of the cell. So you can consider this interface. The outline, short outline, very, very short introduction, because these two problems to put in a same introduction will be kind of more complicated, and then I will describe two different problems. Membranes of cells, pattern formation of the membranes, and a structure of the interior of the cell, cytoplasm. And I will finish with a short summary. Pattern formation in cells. I was expecting actually Ernesto Nicola here, but for me that he left. Actually, he's part of this work, or one of the works after that. You have here an embryo cell, so it's a cell, and before the division, in order to form the organism, it already makes or produces a pattern, which is basically a simple pattern, but it's a static pattern with one part, it's a polarization of the cell. So you need some proteins which attach to the membrane one side, and other proteins attached on the other side. So it's the first pattern formation example I will show here, and it's a very characteristic one, and you have the interaction with the membrane, with the interior of the cell, and you have all the dynamics, and probably you have heard some talk of Ernesto about this. These are oscillations in bacteria of a certain system of proteins which oscillate. They have a clear oscillation inside the bacteria, so they accumulate in one side and they go to the other side and go back. So this kind of oscillation in such a way that they stay longer in the both sides of the cell, and in the middle they are less, and they allow the accumulation of different proteins which will allow the division of the cells. So it's a different example of pattern formation inside of the cells. Cardiac alternance. Here you have a myocyte, which is a cardiac cell, which contracts. And in the contraction is related with calcium oscillations. And if you follow the size of the calcium oscillation, actually this color is the size of the calcium oscillation between one beat and the next beat, the next contraction of the cell. And the size could be different, as you can see, red and blue. So you have a large oscillation, a large concentration of calcium, a beat with a small concentration, a large concentration and a small one, but also it can be mixed. So here the increase of concentration is localized on one side, and you have kind of a standing wave, something like that. And it's not like an alternance in terms of cardiac dynamics. You have in neutrophils also chemical propagation, chemical wave, which propagates, which oscillates. Also, you have accumulation at the membrane, and also, like in this case, the proteins accumulating in one side will produce some effects on the membrane, on the tension of the membrane, which can produce a path. For instance, this is also associated. Here is a phenomenon, as a pattern formation phenomenon, also here. Dictyostelium discoideum at the membrane also accumulates some proteins, which actually this blue, this is certain proteins accumulation, and this is blue is the accumulation of the F-actin, so the part of the filaments which push the membrane. So you have now motion in that direction, where you have more accumulation of actin. And it's due to, or it's very correlated with accumulation of the proteins at the membrane. So it's again a pattern formation phenomena which can give rise to motion. So more classical example is calcium wave in oocytes. you can see very nice spiral waves. These cells are huge, also by the way, so you can see a lot of pitches of the spiral wave. In particular case, the cells are very large. And this is also quite recent, but is slightly different. This is not a chemical pattern, it's a distribution of forces or stress into certain cells, because it has some relation later with what I'm going to speak in the second part. So the stress can be also located, how strong is the force at the surface of the cell when it's moving, for instance. Also, the elastic properties also can differ from one side from one point to the other. Okay, that's kind of an introduction, general introduction, that in cells, inside the cells, you can actually have a lot of nonlinear phenomena. And this will be the first subject. I will focus basically at the membrane of cells. And in order to do this, I will begin with a very generic problem in biophysics or biochemistry, actually, which is the phosphorylation of proteins. Basically, if you have a protein, a phosphorylation of a protein is to put a phosphate. And it's a chemical reaction done by enzyme. You put a phosphate, and then you make this reaction. And the phosphorylation is to remove this phosphate. And this is controlling millions of processes in the cell, these types of reaction. Of course, the phosphate is taken from the ATP, which you know is the chemical unit of energy. So, and then one can think that, for instance, this MARCKS would be a protein, particular protein, free protein, phosphorylized protein. You can think at the very beginning, a very simple description will be kind of this, very, very simple. No? Of course, I say that this enzyme produces a reaction, so it's actually not true. You cannot describe in this very simple description. You need to use Michaelis method, for instance, description. So you consider a kinase, the technical name is kinase, which phosphorylizes, which in this particular case is a particular kinase, protein kinase, which goes to the protein, interact, stays for a short time like this, and introduce a phosphate. And then after that, a phosphatase will remove this phosphate, it accumulates for a certain time like this, and then remove the phosphate. And if you assume that this is the cell, this is the membrane, and you have kinases and phosphatases inside of the cell, you can arrive to some equations using Michaelis-Menten approximation. And you have again the free proteins and the phosphorylized proteins. And they are correlated, they are related, so you have this reaction will depend on the concentration of the kinase, and this reaction, so this one will depend on the concentration of the phosphatase. And this kind of model was also a classical description of this kind of equations, a classical description of phosphorylation, dephosphorylation by Goldbeter and Koshland. And then they were describing these dynamics, and basically they show that with this kind of equation you have basically a switch. So you have a particular concentration which a very fast change from two low concentration of phosphorylized proteins and very high concentration of phosphorylized proteins happen when you change one of the parameters, and this kind of switch. So you change a little bit, then you go from one side to the other. But they are never able to find this stability. So if you don't have this stability, you are not going to have pattern formation in general here. So you are not going to have pattern formation with these kind of equations. Are too simple. Our idea was to consider certain processes where you have to take into account that there is membrane, and that the kinases are associated to the membrane. If the kinases are associated to the membrane and the phosphatases to the interior of the cell, then you have processes at two different local places. You have the protein have to go to the membrane, then it's phosphorylized, and when it's phosphorylized by PKC, which is in at the membrane, then it leaves the membrane, which I will describe why, and goes to the interior, and interior will interact with the phosphatase. It will be free, and if it's free, have some tendency to go to the membrane again. And you have a cycle. And now you consider three types of concentrations: the proteins of the membrane, the proteins phosphorylized, and the free proteins. And then you have a more complex situation. You have three concentrations now. I have actually reduced a little bit the effect of the phosphatase, so it's a little bit simple, the effect of the phosphatase. And then I have again the effect of the kinase, but now have to be attached at the membrane, the protein kinase. And there are some prefactors which takes into account if the membrane is full or is empty. Also, some kind of penalty term if the membrane is already full. So the proteins cannot go into the membrane because it's full. And with this, you can, it's basically the same expression for but in a one-dimensional system. So you can consider a very elongated cell, for instance, or you can go to 2D, will be equivalent to 2D, but it's simple to show in 1D. Then you have the diffusion of proteins. Also, an important ingredient also is known that the diffusion at the membrane is much more smaller than the diffusion in the interior of the cell. The interior, the proteins can diffuse faster. I mean, the membrane is not a solid; it's also a kind of fluid. It's a two-dimensional fluid, but the diffusion is much slower.
If you have a one-dimensional cell, how do you distinguish internal and external part of the cell?
You can basically you can consider a one-dimensional transport into through the one of the phases. You can consider kind of a B model. So two compartments model, but 1D. And you can do it in 2D. Basically, it's the integration of the cytoplasm. But this can be extended to 3D. So I actually did it, but I'm not going to speak too much about it. But the difference is that there is more dilute, so you need to increase the rates in some way to avoid this dilution. But the effect persists in 3D.
So your DM, D, and D are spatially dependent? Or
no, are constant.
Constants?
Yes, but one, because this M is at the membrane, so it always moves at the membrane. And when it moves at the membrane, the diffusion is very low. And when it moves in the C, it will be cytosolic protein, so free protein, and phosphorylized protein.
But so those are two constants, DM and D?
Yes, DM and D is constant. The diffusion will be constant.
Funny way to write the equation.
Yeah, because I mean this comment is a very general. It's a flux.
And do you consider also, Sergio, that the M plus C, the membrane form and the cytoplasmic form is constant?
Yes,
or?
yes, and that's important. Yes, you're right. It's the total number of proteins will be constant, and actually in this phase diagram, T will be the sum of all the protein or the integral. And in more complicated geometries, have to take into account the difference between the membrane and the cytosol. And for instance, as you can see in the phase diagram, already changing one of the parameters, which is the reaction term. So how PKC can phosphorylize the proteins of the membrane. So how fast the enzymes can remove the proteins from the membrane. Then you have typical pattern formation in terms of equivalent to Turing pattern, because here this diffusion is much larger than this one. It's not Turing pattern. Well, it doesn't look like a Turing pattern because there is a conservation law, I should say. So there is coarsening at the end. So, but there is a particular wavelength which begins the instability, and then there is coarsening.
You still assume that the membrane and the cytosol have the same dimension. So the membrane.
Yes.
So it's not a 3D, a 2D involving a 3D ball.
No, it's basically you project in order to do this model, which later I will go to a more complicated model. But in order to do this model, you integrate the whole. You consider a kind of thin layer of the cell close to the membrane, and you can integrate in the vertical direction, and you assume that there is no pattern in the vertical direction, which can be relaxed later, and I will show that it doesn't depend on it.
What did you say that there was the relation between coarsening and conservation?
I mean, the conservation makes that in K0, the instability begins in K0. So in K0, it's equal to 0. And typically, coarsening is always observed when under this condition. When you have a maximum which begins from K0, goes to a maximum, and goes to 0. And this coarsening is always associated to this kind of process.
Disagree.
You disagree? Okay. We can discuss. But coarsening is always associated. I mean, you have a...
You can have coarsening with no conservation.
Maybe, but then the property would be
Kinetic
different.
acid.
No, you can't.
Yeah. Yeah. Could be, yeah, I mean... Okay. And yeah. And basically, if you cut this phase diagram from here, you have the solution, which is the very few proteins at the membrane, and then it becomes unstable due to this instability, and then stabilize again to when it saturates. Basically, the membrane is saturated. When you increase, this is the total number, or the total number of proteins. So you increase the number of proteins, and then at the end you are going to saturate the membrane. And at the beginning, there are not enough proteins to do anything when they stay outside. And in the middle, there is instability. And if you go in that direction, also there is
instability of what?
The homogeneous state.
Homogeneous state.
Yeah, sorry. To calculate the stability of the homogeneous state, from these equations, you arrive to also to P stability.
Yes.
And here is typically associated to P stability, and together with the conservation law, also give rise to a pattern formation with different properties that in this side, in the size of the domain. Yeah, for instance, simulations in these 2 points here from a random initial condition, you have the generation of pattern formation with a characteristic length, and in this position of instability for a homogeneous with a little bit noisy initial condition, it remains in this homogeneous solution, or if you put at this kind of initial condition and a step initial condition, you are
That's time or x?
Sorry, that's x. Sorry, that's
a snapshot.
it's a snapshot at the end of a numerical simulation.
Okay.
Sorry.
It's not the final step.
Or the final state, yes. You can run later much more numerical simulation, but it's a final state of the particular numerical simulation. Here, for instance, will be coarsening, so it's not a final state, but it will take. It's a 1D also simulation could take forever. But here, depending on the initial condition, you are going to stay also coarsening. You are going to stay pattern formation, which is stable. It will be this is the final state.
But do you impose boundary conditions there?
It's no flux boundary condition.
But why the two states are equivalent at completely identical stability? Why one of the states does not invade the other?
Because the conservation law. It's a little bit, you cannot think in terms of a typical stable system. If you have a conservation law, you have this kind of solution which appear. It takes energy to remove this border. So it's a minimum of the energy. It's one of the minimum. It's not the the most stable situation. The most stable situation probably is in a homogeneous state. But depending on the initial condition, the situation is metastable, but relatively stable. So in this kind of two different mechanisms of pattern formation. Well, to start. Okay, then I go to a particular example. It's the MARCKS protein, which I will try to apply all these calculations, much more theoretical, to this particular protein. And the particularity of this protein is a kind of an elongated protein, so it's quite small, and has some affinity, because it's charged, the protein. So it's actually not hydrophobic, or not completely hydrophobic. There is a part which is hydrophobic, so it doesn't like to go to the interior of the cell, but the protein itself could stay in the interior. It don't have any problem. It's not globally hydrophobic, or not enough to go to the membrane, but have a charge which interact with acidic lipids, which are lipids with a different charge, and they go to the membrane and interact. The point is that the characteristic of this protein is very high concentration, in particular in neurons. Neurons have a huge concentration of can arrive to 10 micromolar, and it's unfolded and hydrophilic membrane protein, which is a little contradictory. And it binds to the membrane because of this effector domain, so electrostatic interactions with some phospholipids, and this part also will help, but it's not it's not enough to keep the protein attached. And it unbinds from the membrane by phosphorylation by protein kinase. So basically, a protein phosphorylizes some of this charge, and then there's no charge anymore in the protein, so there's no interaction with the membrane. So it detaches and go to the cytosol. And there is activity by calcium and other stuff. If you have a cell, basically you can perform an experiment where you bin PKC with red fluorescence protein, and if you don't have any increase of, you have a normal conditions, the PKC will be at the cytoplasm. But this PKC is activated by calcium, and if you have a calcium spike, a calcium increase of the concentration can induce the activity into the PKC, so it means that it have affinity to go to the membrane and then accumulates at the membrane. Then marks typically are located at the membrane, and when they peak after a calcium spike, so the PKC goes to the membrane and then interact with the marks, and the marks go to the cytoplasm. And then kind of more experimental example, so you introduce a chemical and it induces calcium spikes, and if you are visualizing calcium, if you visualize PKC, the same chemical induce also this decrease of cytoplasm PKC concentration. So when you go down, it means that in the cytoplasm there are less PKC, it means that it goes to the membrane. And this is also the concentration in the cytosol, and this is in the membrane, at the membrane. So increase at the membrane, and they extract from these kind of pictures.
So this is the second. The lower plot is one of the spikes.
It's one of the spike. It's one of the spike. Okay. Of these spikes. So the red one is the concentration in the cytosol. So they calculate here, and the membrane they calculate here. Increase the concentration of this enzyme, and at the same time they can look in the cytosol the concentration of PKC and of MARCKS. match now with the green fluorescent protein, and they see that in the cytosol, when the PKC decrease, so it goes to the membrane, in the cytosol, the MARCKS proteins increase. So it goes from the membrane to the cytosol. And one can consider a cell, model these kind of processes. One has to consider unbinding binding of PKC, which is activated by calcium, and phosphorylation when it interacts with PKC. It's phosphorylized, it will be phosphorylized 3 times, and the phosphorylation and binding and unbinding of MARCKS proteins by themselves. One can try to make a numerical simulation in a circle. So you remove some product, which is the nucleus, and you consider a slice, which is the membrane, with some particularities, which are different than the particularities on the other slice, and make a numerical simulation with different diffusion coefficients, which can be estimated.
Sergio, but,
yes.
The simulation is with, here is with the initial model, right? I'm asking because there is a thing that you say, typically this phosphorylation, you have the kinase and the kinase, but each one can have in principle different constants, different affinities. So you are in a way considering just as one effective...
I will do numerical simulation of this kind of model. So I consider only the PKC, this type of enzyme.
Okay.
This type of enzyme and this type of protein.
Yeah.
I'm not considering also, for instance, stochastic
the MARCKS,
because
MARCKS to match with one
with phosphate,
with 3.
Well, basically I reduce quite a lot, and then I consider it with 3 phosphates and the binding and binding of MARCKS proteins. And this is a particular phosphatase, PPA. It's one particular phosphatase which is doing this job. Stochastic effects actually can be included, basically because the number of phosphatases probably is not very, the concentration of phosphatases is not very large. Enzymes will be larger, but not so much. But MARCKS proteins have a large concentration. Here we can neglect stochastic effects. And basically these results, I will show only the results, just fit basically some of the parameters, and it's not also trivial. It's always say that with a couple of parameters you can fit everything. Sometimes it's not true, but I mean you can arrive to fit somehow the experimental data, which are the points with the lines, which are the results of the numerical simulations. And basically how it works in the experiments. So you have the high concentration of PKC at the cytosol, and then it goes to the membrane. The MARCKS protein goes to the cytosol. Then PKC goes back to the cytosol. In some moment, it's already back to the cytosol, but this is still the protein. The MARCKS proteins are also still in the cytosol. It forms out some time scales also, what is the process which is faster, and then at the end you recover the initial state. And doing these numerical simulations in a circular domain, mimicking one cell, we actually ran because we knew some results previously, we ran numerical simulations for a long time. And then at the long time, just checking with the same parameter value which I was using to fit the experimental curve, you arrive to this is the perimeter of the membrane, and this is time, and you arrive to domain formation, pattern formation, the final state. To the MARCKS proteins are localized in one side, which produce the polarization of the cell. And with a step initial condition, with changing in the phase diagram, you can reproduce the phase diagram with a little slightly more complicated model, and you can go to the place of V stability, and you recover again the V stability here, which depends on the initial condition. The homogeneous state is also stable. And that can be And then we have some collaboration with a Japanese group and trying to do this, to try to look for longer time and try to stabilize the cells in order to find the tuning patterns. We have some, we try it, and they are trying to work on it. I don't know if we come back with some results. And also we have some collaboration with experimentalists in Leipzig, with Professor Joseph Kess, and they were considering also the similar problem, but in vitro. So not in vivo, but in vitro. And a similar model of a cell is basically you have water and a monolayer, reduced system of cell. And now the membrane is just one monolayer, and you can put in all the products you need in order to keep stability of everything. You introduce the MARCKS protein in the bulk. After some time, it accumulates at the monolayer. Then you introduce the PKC, and the PKC also go to the membrane, and the MARCKS are removed. And you can measure actually indirectly the concentration of MARCKS at the membrane, because the lateral pressure of this monolayer without and with proteins is different. So you detect a change on the lateral pressure, which can be measured very nicely in the experiments. So you see that it's changing and then it's removed. And actually what they observe is some kind of oscillation, which we will have to adapt our model to this particular geometric configuration. The conservation is a closed system, so they have oscillations which disappears with time. Because also there is ATP, of course, dissolved here in order to produce the reaction. But we have temporal oscillations. Of course, they disappear because of the closed system, and we more or less reproduce them. So there's a different type of experiments in vitro experiments, and we try to also develop the model to this kind of systems. Okay, that's one part. I mean, I hope that was more or less clear. Stability, pattern formation, and the membrane, the mechanism was basically produced by the difference of diffusion coefficients at the membrane and in the cytosol, and some nonlinearities and the conservation law. Here is a completely different scenario, and we are going to consider now the cytoplasm and viscoelasticity also. What is typically with AFM, they go to a cell which is living here, and they perturb with a cantilever, and they can measure very nice the force of the responding to this force you are putting in the cell. So you are calculating the elasticity of the cell, basically the cytoplasm, and producing, for instance, oscillations. You can have a large spectra where you can calculate the viscoelastic properties of the cell. But on the other, and then there is some kind of dilemma if the cell is a viscous system or is a solid system, and under which perturbations. Basically, for short perturbation, the cell behaves as a solid, and for large perturbation, it's like a fluid. And also, additional information: cytoplasm is formed by two phases: a cytoskeleton, or a solid plastic meshwork. So it's basically the cytoskeleton together with other macromolecules, and a fluid phase, a cytosol, a fluid. So you have a model of a cell is basically a sponge. So you have a fluid inside of a network of fibers, of actin fibers. And how can they evaluate that is actually a poroelastic system, it's a porous system. It's a very simple experiment. They introduce small spheres into the cell, so it's very small, and depending on the size of the spheres, they diffuse through the whole cell. This diffuse is lower, as you can expect, because the size is larger, so you have to expect that the diffusion will be lower. But if the size of the bits are large enough, they are trapped, so they don't move anymore, they don't diffuse. And this is one of the experimental evidence that there is a particular characteristic size in the network, and they can estimate the characteristic size of this network by these experiments. 20 nanometers, something like that. And different experiment is that you have dehydration experiments. So you have a normal cell here, it's nice, and you, by osmosis, or localized osmosis, you can dehydrate, you can remove the whole liquid, the whole fluid from there. But if you expect a fluid system, if you impose a pressure, a difference of pressure in one side, so the whole fluid will go to these sides and will disappear. But if it's not going the whole fluid here, the cell doesn't feel anything. So it's a confirmation that you can describe this as a poroelastic system. The poroelasticity has some particularities which allows gradient of pressure. So there was kind of two different experiments in order to check the poroelasticity of the cytoplasm. Okay, poroelasticity is a mechanical property, but we are basically interested typically in biochemistry. So how the reactions appears, how are the non-linearities of the reaction, and typically the biochemistry can alter the dynamics of the molecular motors, can change viscosity, because can change the chemicals involving, can change the mechanical properties of the cell. But on the other hand, the mechanics, if you change the mechanics locally, you can produce transport or induce localization of some of the components of the system, some of the chemicals which are inside, which are diffusing in the cytosol. And now there are a lot of works now in active gels. So the group of Julica now is working a lot on this kind of active gels and how can relate mechanics with activity, with nonlinear reactions. And then we put together both concepts, active and poroelastic model. So we have a cytoskeleton, which is a solid phase, which inside there is a fluid phase. So they are interpenetrating phases. Then both phases are coupled because this is a sponge phenomena. If you make some pressure in the filaments, then the fluid will leave, and it's a sponge. So they are coupled. You have cytoskeleton and you have cytosol. We consider for the moment a homogeneous distribution of molecular motors. Molecular motors are proteins which are in the filaments, and they are making forces. So they bring together. They make a kind of homogeneous tension in the cytoskeleton. We have a tension. It keeps the cytoskeleton all together and makes forces. And we consider a regulator because the molecular models depends on some regulators, some ions, which can be transported by the cytosol, by the fluid phase. And this regulator modifies the properties of these molecular motors in some way. Okay, and also some steps in order to derive the model. So we consider three elements. So the deformation of the solid. So how can we, a solid if it is deformated, how it moves, or the compression of the deformation of the reference state, if you want. So it will move because the pressure of the motors change, local change. Then if you contract like this, there will be some fluid which leaves from this part and goes to the empty space. So there is a fluid, which is the cytosol, and there is a concentration of some biochemical which move with the fluid. Then we consider incompressibility, which some particular boundary conditions can be integrated very easily. I will derive everything in 1D because it's much more clear and the equations are more simple, but they can be extrapolated to 2D, and we have already also done in 2D, but then it's a bit more messy and more complicated, but the concepts are basically the same. And the stress, you have to consider stress in both phases because we are speaking about a B phase model. This is the stress in the solid phase, which is the effect of the elasticity. This is the Young modulus or the elastic modulus. The effect of a dam, the viscous dam, in again this deformation. So this is viscoelasticity, and then this active tension. So it's active produced by the molecular motors, which are add to this solid phase. And the fluid stress, which is basically a viscous fluid, so the viscosity of the fluid. And then you apply force balance with the Darcy's law because it's a porous media, or you can see as a two porous media. And we go to the body reference velocity. It's kind of a change of reference frame, which will allow to reduce a little bit more the equations. So we arrive to a two-phase model, which is the fluid motion with incompressibility. This is the regulation. Actually, will be the solid motion more than the regulator. Well, both equations are for the solid and for the fluid because they are coupled. And this is the equilibrium state, the force balance. And this is the dynamics of the regulation. There is an attraction term due to the velocity and the diffusion. There is always diffusion. And for the moment, we don't consider any reaction. The only reaction will be here in the active tension. And I will explain now. So this term comes from the viscoelasticity. The active tension is this one, which depends on the concentration which is affected. So it controls. There is a homogeneous tension in the whole system, and it's locally reduced by the effect of this regulator.
Can you explain more? What is this regulator?
I will go to experiment and will be calcium. Okay. In myocytes, for instance, calcium activates the stress, but there are some organisms where the calcium inhibits. In other systems, also magnesium can be which the concentration is very low, but it is also an inhibitor of the molecular motors activity. And then you apply here the Darcy's law and its porosity also because the incompressibility, and we introduce Darcy's law here. So you can calculate the permeability, which is directly related with the size of the porous. So the size of the porous will appear into this parameter actually, and the viscosity of the fluid.
I don't know with the molecular motors, how will that work? I don't know, you say you also have molecular motors.
Yeah, the molecular motors are making this tension. This tension, the active tension, are done by the molecular motors because they are putting those two elements and they're keeping together, or they're moving typically the filaments. But I consider the molecular motors are homogeneously distributed. You can consider also dynamics in the molecular motors, but first approximation, but they are affected by some regulator. So sometimes they need some activator, or they have some inhibitors in order to make the force or the the stress they are doing depends on some chemicals. Porosity, so again, the deformation, the sponge model. So there's a deformation of the solid and the velocity are coupled. There's a regulation will move in the fluid and affects basically by the active tension the deformation of the solid. We can make the stability analysis of this model, and we have wave instabilities. It's a different type of wave instability because the maximal is real, but then also some temporal dynamic will observe. I'm not going to show the numerical simulations. And also you can arrive to this part when there is not elastic force anymore, and then you have a static pattern formation, which connects with the limit of Stefan Green on his paper of active gels, for somebody knows about it. And I will apply this to a particular organism, which is the Physarum, and I will try to go very fast. It's a very huge cell. It's a cell with multiple nuclei. But the nuclei are always the same nuclei, well, the same in genetic information. And basically, from this base, one can extract a little bit of fluid, and if you put the fluid apart in a petri dish, a droplet of this microplasmodia, so a part of the plasma, then it begins to oscillate, but physically oscillate. So you can make experiments and check the thickness of this droplet, and the thickness of this droplet is changing. And then it's oscillating, for instance, or there is a wave, can observe a standing wave, and this is thickness, it's not a chemical system. And global oscillations. Also, it's known that it's coupled to a chemical oscillation of calcium. But all this deformation and thickness will induce also flux. And basically, we applied the model, but now the chemicals are two chemicals, and they are coupled with a reaction. In this case, it's the brucellator. One can go to a more complex models of Physarum, but for the moment, we keep in the brucellator, which is giving oscillation, which is what the calcium is giving rise. And you have again the same, but now with a oscillation here. And you obtain again wave. These two types of wave are basically with a maximal imaginary of the maximal real is have an imaginary component or don't have or doesn't have an imaginary component. But there are some part of unstable imaginary parts. And then you have also wave, and there are some typical numerical simulations that also can be done in 1D, also in 2D can be done. Here is 1D for the wave. This will be the deformation field can be estimated on one dimension how it will modify the thickness. Or can be some kind of argument, and the chemical wave of calcium. And also for you can see here wave and standing wave. So with these simple models. And the summary. So I think it's quite late. It's already 1 hour, so it will finish quite fast. So there's basically what I said. There's the summary of what I said about the modeling pattern formation of the membrane and the poroelastic model, and then some scientific collaborations. I was working in the group of Marcus Veda, have experimental collaboration with Professor Kies, with Karin Joning in Grenoble, also with the people of the TU, and with the experiments also with people in Magdeburg, Professor Hauser. And funding, also PTV, also paid some several things, and the SSF is a kind of program or project in Germany. And that's the list of references, and thank you very much for your attendance.
Was you want to say?
I was the chairman. Questions, comments? Is this
I don't know very much about those active gels and those the coupling between biochemistry and mechanics. It's an active field. Do you know of any? I'm sure that being in this PTV, you should have some information about possible applications of this for small devices. Do you have any idea?
For the small devices.
Well, I have heard something, but I don't know.
I didn't heard about it. I mean, probably. I mean, the point is that you can. I mean, at that level, you go to the microfluidics. Maybe you can try to use these mechanics to produce fluxes. I mean, that's also something that I was working some time ago. Also, the idea of control microfluidics by active biochemistry or active gels or active something. So it means that with energy you can produce fluxes, at very fluid fluxes, at very short scale. And it was kind of interesting in order to control, but I don't know any specific application in terms of technology to this kind of processes. We are more interested in modeling the processes in the cell, or how they basically begin to move us would be the next extension. How can these deformations inside of the cell produce fluxes of fluid, will push the membrane and will produce some kind of motion. They are typically using these poroelastic models or two-phases model, basically. The two-phase model to model the generation of protrusions in the membrane, in order to how to model when a cell begins to move from this kind of motion.
More questions, comments, Manuel?
You were saying at some point that in one of, the second topic, you were saying at some point that in one of the limits, that corresponded to the Stefan Green's results. Which are exactly the new ingredients that you have? Is there a regulator or the calcium or the?
The new ingredient is the viscoelasticity. So I reduce. They are not using viscoelastic. They don't have a viscoelastic system. They have just a pure diffuse system coupled with an active tension. And in that limit, it's a viscous system, some kind of viscosity, but the elasticity is not incorporated. And in that sense, the reduction in this direction, when I was reducing the lasting model, you could arrive to this limit. Also, the approach of them is slightly different than our approach, because they consider the motion through a diffusion in the solid phase, which is not solid for them. There is a gel phase, and then it's activating the molecular motors. We have the transport through the fluid phase and the inhibitor in such a way that there are two different signs which compensate and give rise to the model of Stefan. Not the same results, but the model are different, but they compensate the two signs. So in that sense, they're reduced, but they are not equivalent. Also, in this limit, the approaches are coming from two different systems. They consider active fluids, basically, not active solids.
I have a general question. You've shown us two examples of pattern formation in living cells, and in each of these examples there are a number of interesting questions that you address. Now, is there what is the context? What is the general question for which pattern formation in living cells is interesting?
Why? Okay. One have, in order to understand, basically all these kind of things are addressed in order to, first of all, the first model will be more in the direction of polarization. Polarization is important in cells because it produces the direction of motion. If the cell is going to move, it will first of all polarize, and then it will move in some direction. So understanding how is the process to pattern formation inside of the cell, you can basically understand why it's moving in one direction or not in the other. Also, as I show in the first slides, also when there is division of cells, this begins sometimes with already a pattern formation in the first cell. So actually, these cells divide. This in the first example, when you have a membrane accumulate and protein accumulation in one side of the membrane, and the different type of protein accumulates in the other side of the membrane, it divides. And basically, in one cell will be only one protein, and in the other protein. So it will develop completely different parts of the organism. And understand these processes at the scale of the cell. I think that is for general context. For general.
I guess I would rephrase Max's question. What's the crucial questions people are trying to answer?
The crucial questions?
Crucial questions. I mean, you can clearly find many examples of pattern information in cells. Spend your life doing it, but what are the crucial questions you're trying to answer?
Going to a more philosophical time of questions.
No, not philosophical.
Oh, no, philosophical, no, in science, yeah. Is to understand the cells at the scale of the cell. I mean, what are they trying to do, and how are they using the chemistry, how they are using the non-linearities. I mean, you can work a lot with biology, and you will understand a lot of things in cells with biology. But in some moment, you need to incorporate non-linearities, introduce pattern formation, because what the cells are doing is not only described by biology, also by physics. And why the cells are moving in some direction, why the cells are controlling. I mean, you have all this field of system biology now. They have only local reactions, and they are very good, and they have thousands or not new results. But in some moment, they have to try to go to spatial. You know, it's not the same that the reaction happens at the membrane that happens in the nucleus, and it induces a delay, induces gradients, induces these kind of things. And in this context, non-linearities are important.
I could understand saying that understanding cell motion is a crucial question. Yes,
it's one of the main questions, also cell division also.
So I guess my question is that the direction your research is focused on?
Yeah, it's going to the direction of motion, to the motion of cells. That will be my research. Actually, we are going to begin now with master student, also in this too. How can we couple to the motion? How can we choose first of all a gradient, and then how the cell will move in some direction and why? And then there is a coupling between mechanics and chemistry or biochemistry. It's important.
My question is, to which extent the pattern that, for example, you obtain, or the dynamics that you obtain, is determined by the boundary conditions and all the mechanisms that you set there to the very beginning? I mean, so you are there a detailed description of the chemistry and interaction, and then you are there the boundaries and everything.
Yes.
So, to which extent the pattern formation is determined by non-linearities, or this is just an effect of boundaries and all the ingredients of the system?
I mean, of course, everything collaborates into the formation of pattern, but the boundary conditions are typically not very relevant in order to induce the pattern. Also in this second part, it's very crucial that you have no flux boundary conditions into the chemicals and constant, because you consider a circle. But of course, the next step also will be the motion. So allow these different boundary conditions, which does not affect the pattern formation itself, but it will affect the response, it will modify it. And how the cell will begin to move in one or other direction through the nonlinear properties of the reaction. There are multiple of things we have taken into account, of course. But and then if you want to describe a whole cell, then you have to go also to three-dimensional systems and to consider very complex bioelasticities.
Okay, so if there are no more questions, let's thank Sergio again for the presentation.